Hello, my lovely anatomists and physiologists. Thanks for joining me for this topic of red blood cell recycling. So remember when we talked about our red blood cells, we said a mature red blood cell is lacking most of the organelles. And so this means that, as they're rushing through our cardiovascular circuit, moving from arteries to arterioles to these super tiny capillaries, which they squeeze through, and then back into the larger venules and even bigger veins and back to the heart, we see that they can accrue quite a lot of damage in them moving through the circulation, and then they lack this ability to repair, right? There's no nucleus with an instruction to make proteins. So these cells have a very specific sort of time frame of about 120 days. And what we see is as these cells begin to kind of get, let's say, old and worn out, they lose their flexibility. And I'm just going to say, like, they hold on to their oxygen. So, their whole purpose is to move around and transport oxygen to your tissues as needed. And, if they're losing their flexibility, they're kind of clogging up the passageway. And if they're then holding on to the oxygen, they're not delivering it. And so they're pretty much not fulfilling their purpose. And so when this happens, we see the cell called the macrophage play an important role here. And so the macrophage, I'm going to draw like a Pac-Man. And I'm going to give it big teeth. So the macrophage is this really big phagocytic cell. It's actually derived from the monocyte, which we've mentioned before. We'll mention again as one of our white blood cells. And macrophages are going to be super important as phagocytes in the body. And we'll see that they can roam around in your tissues, just kind of loose and eating stuff that needs to be eaten. But they also are going to be like living in the spleen. And they're also going to be living like in the liver. And we also see them living in the bone marrow. And so what we see happening is as your blood is moving through these different parts, these macrophages have this ability to detect like, oh, that red blood cell is getting old and we need to remove it because we don't want it to grab hold of the oxygen and not let go. We don't want it to clog up our blood vessels. We need to make room for the new functional cells. And so the macrophage is going to come along and gobble up and recycle key components of the red blood cell. So a lot of this is happening actually inside the macrophage. So let's designate that here. So we're actually inside the macrophage. And we're going to see the following things happen. So we're going to break down the hemoglobin molecule, which I'm going to abbreviate here as capital H, lowercase b. And we're going to be able to, I'm going to say, reuse the amino acids. So we can see the macrophage will break down the subunit of all 4 subunits, the amino acid components of that hemoglobin protein. And those amino acids then are in circulation in the bloodstream, and they can make their way back to the red bone marrow in order to be used to make new. So when we think about like what's actually recycled, we're going to be recycling the amino acids. When we break down that hemoglobin inside that macrophage, we're also going to release the iron from that heme group. Now, iron, remember, is a trace element, and it is the piece that binds to the oxygen. So this is kind of precious in the body. We don't want to lose this in any way. And so we're going to see that this is going to combine with a transporter called transferrin and make its way back to the bone marrow to be reused in the making of new hemoglobin. We can think about that for amino acids too. So if we want to write that in for amino acids, say they're going back to the bone marrow. So we're also recycling here the iron, right? The hemoglobin molecule really is made of three things, right? So the hemoglobin molecule is made of amino acids. That's the protein subunit component. It's made of iron, right? And then we see it made of the heme. And the heme, I'm going to put it in orange, is not recycled. So over here I have this little trash can in my recycling diagram, and the heme is actually trashed. So amino acids are recycled, iron is recycled, heme is trash. Heme is trash. And so what's going to happen inside, we're still inside the macrophage, the heme is going to be converted to a molecule called biliverdin. And then this molecule is broken down further, converted further into bilirubin. Now the bilirubin is going to leave the macrophage, go into circulation, so into the bloodstream. And then we're going to see that at the liver, we're going to get that bilirubin converted or actually used to make bile. So bile is a chemical that's made by the liver. It's stored and concentrated in the gallbladder. And it's going to have a special duct, it's called the bile duct, that's connecting the liver and the gallbladder to the small intestine. And the bile is going to be secreted into the small intestine to help us digest fats. And so the bile makes its way into the small intestine, and then it makes its way into the large intestine. And inside the large intestine, we have bacteria. We have little bacteria buddies. And these little bacteria buddies are going to break down this molecule further, and they're going to convert it. There's two different types of molecules. We're going to get Hello, my lovely Okay, so the bacteria buddies in our large intestine are going to convert that molecule into urobilinogen and then further convert it to stercobilin. And then we'll see this just release from the body in the form of feces. Now, sometimes this bilirubin molecule in the bloodstream gets taken up by the kidney. And sometimes some of this urobilinogen and stercobilin gets back into the bloodstream and gets taken up by the kidney. So what we'll see is that this heme is this kind of pigment of our hemoglobin. And so when we look at these different molecules, we'll see that the biliverdin, verde means green, so this actually has a green color to it. And then the bilirubin, we'll see, has sort of a yellow color to it. And so by the time we get into the large intestine and the urobilinogen and the stercobilin, we'll see that this is going to give some of the sort of brown some brown color that we associate with fecal material. And as I mentioned, sometimes these chemicals make their way and are removed by the kidney. And so then that's giving some of the yellow color to our urine. And you maybe have seen some of these colors in a bruise before. Or if the liver is not functioning in order to remove this bilirubin, you might see this yellow color showing up in the integumentary system. So you can see this yellow-orange color showing up in an individual's skin, depending on how light their skin is. You may be able to see that more or less easily. You can see it in the whites of the eye that would be showing up for individuals who have a liver that's not pulling out this bilirubin. We call that jaundice. We see that sometimes in newborns. So sometimes newborns have difficulty processing their bilirubin. And in that case, we usually just maybe put them in a sunny window for their nap, and that helps to process some of that out of their integumentary system. Sometimes you have to do special light treatments for that. So again, as we're looking at that hemoglobin and we're talking about this removing of these old worn out red blood cells, we see that the macrophage is going to break down the hemoglobin, and there's three key parts, and the amino acid and the iron parts are both recycled and reused by the body, making their way back to the bone marrow to make more hemoglobin. But that heme part is a waste product that is removed through a series of steps. All right. Up next, let's take a closer look at leukocytes. And as always, take care of yourselves and each other.